Good afternoon, everyone. Today we are going to start a new unit, which is the muscle physiology. Now, in human body, there are three types of muscle: skeletal muscle, smooth muscle, and cardiac muscle. In this unit, we are going to study about the skeletal muscle. What are the salient characteristics of a skeletal muscle? Salient features means the unique structural and functional features of a skeletal muscle. Of a skeletal muscle fiber. Now, a skeletal muscle fiber, it is striated. This is the first and the foremost characteristics that we see, physical characteristics. Like whenever we study a striated skeletal muscle under the microscope, we know that it is a skeletal muscle. Second, it is multinucleated. It has got more than one. It has got many nucleus. It is rich in mitochondria. It is rich in mitochondria. That means it is very active in metabolism. The skeletal muscle, it has to undergo a lot of metabolism. That's why mitochondria is more in number to supply with energy in form of ATP. Now, organelles like Golgi bodies, endoplasmic reticulum are also present. Skeletal muscle also stores glycogen. So, glycogen, which ultimately provides glucose for energy, glycogen and oxygen storing pigment, which is myoglobin. What is myoglobin? It is oxygen storing pigment, are abundant in a skeletal muscle fiber. These are the physical salient feature of a skeletal muscle. Now, what is the location of a skeletal muscle? The skeletal muscle, it is mainly the muscles which are related to locomotion, related to mobility, that enables us to move or change in posture. So, muscles of the limbs, hand, legs, back, trunk, all these things have the skeletal muscles. Now, what is the origin and the end of a skeletal muscle? Origin, that is beginning, and insertion of a muscle is in a tendon. So, if we draw this one, say this is a bone and this is like tendon. Okay, this is a tendon. So, a skeletal muscle originates like from the tendon. And what is the hierarchy of it? Like what are the layers of it? The outermost tough covering is called as the epimysium. It is the outermost tough covering. Now, inside this epimysium lies bundles of myofibril or skeletal muscle fiber. Now, the structure that encapsulates or covers each and every fascicle is called as the perimysium. And inside each bundle of this myofibril, which is the fascicle, lies each and every individual myofibril, or if we enlarge it like that, it is a myofibril which is enclosed by again another layer of connective tissue, which is endomysium. So this is the hierarchy of a skeletal muscle. Now, skeletal muscle, its main function, the primary function of a skeletal muscle is to undergo contraction. Okay, this is a primary function of a skeletal muscle. It can undergo contraction, which is very much needed for the locomotion and the movement and any kind of mobility. In context to this contraction, the structure that is most important and the structure that is taking part in the contractility is called as the sarcomere. So, sarcomere, it is the unit of contractility. Now, understanding the structure of sarcomere is very, very important. Here, under light microscope, under light microscope, the image, the image of a sarcomere that we get is like something like this. It is seen that the sarcomere, it has got certain bands. One band is called the isotropic band or I band. Then another one is called the anisotropic band or the A band. So there is like alteration of this I band, A band, I band, A band like that. Now it is seen that when the refractive index is studied in this band, it is seen that the anisotropic band, it is thicker. It is thicker means light is not passing much through this band. So this is the darker band, this A band, aniso, light doesn't pass, and this are the lighter band. Okay, these are the lighter band. Now, electron microscopic structure reveals a structure of a sarcomere which is something like this. So, the A band, we start with the A band. A band is like this. It is like darker and the configuration looks like this. So, it is seen that in the A band, there is a heavy chain which is called as the myosin chain. This is the A band. We'll do the demarcation very soon. And on the either side of this A band lies the lighter bands, which is the actin, which is the actin. So the orientation is something like this. Here, something like this. So this darker region, this is our A band, and On the either side of A band, what we have is the I band. Now, centrally, there is a relatively little thicker line that is passing through the I band, is our which is our Z line. This central line is our Z line that passes through the I band. And now in the A band, in the middle of the A band, there is the very thick region, the thickest region in the A band that is kind of bulging out. That is kind of bulging out. It is called as the M line. The single line that we are seeing, it is called as the M line, which is like very, very thick. And on either side of the M line, there is relatively lesser or lighter region, which is called as the H band. So, it is like the area that has the myosin, filament, which is also called as the thick filament, makes up the A band. That's why it is thick, very, very thick, and the refractive index is also like high. It means not so high, it is like darker. Whereas on the either side of the A band, it is made of the actin filament or the thin filament. Now, this region or the area which is between two Z line is called as the sarcomere. It is called as the sarcomere. Now, this configuration that we have studied, like the one just now we are talking about, this is the this is the sarcomere in resting condition, means when the skeletal muscle fiber is in relaxed condition and has not contracted at all. This is how the A band and I band lie, where in the A band there is very slight overlapping of the actin, that is the red lines as you can see, and the myosin, the darker blue lines. So there is very little overlapping, very less overlapping, but no contact, no physical contact of the actin and myosin are there. So actin and myosin are like just lying next to each other without any physical contact during the resting condition or the relaxation condition, when it is not contracted at all. Now, during contraction, during contraction, this whole A band and I band, they come closer. So the structural change in the sarcomere during contraction that happens is like this. So very quickly we are going to draw the A band. Okay. So this is the A bands, that is the myosin filament. That is the myosin filament. Okay. The A band, this is the M line. Now, during contraction, what happens? The actin filament moves very close to the myosin, and they also have physical contact with the myosin. The mechanism we will study in the excitation contraction coupling. That's where we will study the mechanism. So for now, see the structural change in the sarcomere. So during contraction, the actin and myosin, they move closer and they touch each other. So here you can see the distance of the I band from the A band have become lesser. I band, I band. So during resting stage, they were quite far off, right? Now, when contraction started, the I band, that is the actin molecule, it is pulled towards the A band, that is the myosin molecule, and there is more overlapping and physical contact of the myosin head with the actin head, right? So here what happens during contraction at the level of the sarcomere, this is Z line, this is Z line. So during contraction, the length of the sarcomere shortens. The length of the sarcomere shortens. That means the Z line comes closer to each other compared to what it was during the resting condition. Another point that consciously needs to be remembered is that during relaxation stage, what did we know? We saw this sarcomere, it comprises of, see here, half of the I band, all of the A band, and again half of the I band, right? This is one constitution of the sarcomere. So sarcomere, it is like, it has, ultimately it has one I band and one A band, but the orientation is half I band plus one A band plus half I band. So during contraction, the length of the A band is constant. So just see here, the A band, which is like depicted black in color, see the myosin head and the myosin molecule, their length at the sarcomere, that is being maintained constant. It is not reducing, or reducing in length, whereas it is the actin filament that is being pulled towards the myosin. So it is the only I band that shortens in length. So the A band remains constant and I band becomes short. So what is the take of it? The take from this is that during contraction of the muscle, the length of the A band remain constant. The length of the A band remains constant when during contraction. This is the take from the sarcomere, and it results in the various kind of tensions, passive tension, active tension, which will come later. This is understanding the sarcomere. Now, when the contraction happens, another structure equally important like that of the sarcomere is the Sarcotubular system, which is the Sarcotubular system. Now, Sarcotubular system, it includes L tubules, it includes T tubules, L tubule, T tubule, and it also includes triad. Now, what are they? We know that the structures of the muscle fiber, they are named differently from that of a normal cell, right? So, what do we usually call the cell membrane of a muscle, the cell membrane or the plasma membrane of a muscle is called the sarcolemma. It is called the sarcolemma. Just like the endoplasmic reticulum is called the sarcoplasmic reticulum. These are the things that we already know. So, if this is the sarcolemma, that is the plasma membrane of the muscle fiber, from the plasma membrane, that is the sarcolemma, there is like invagination, finger-like invagination, inward projection transversely, inward projection transversely, which leads to the structure, that we known as the T tubules. The transverse tubule, it is called the inward finger-like projection, is called the T tubules. Now, on the either side of the T tubule, there are longitudinal tubules. There are this longitudinal tubules. Now, this longitudinal tubule are called the L tubules. I'm just drawing like one side of it, like half of it. The same thing happens in the other half of it also. What are the characteristics of it? The characteristics to remember about the T tubule is that this T tubule, they have this calcium receptors which are voltage gated. They have this voltage gated calcium receptors. Okay, this voltage gated calcium receptors, which are called as the dihydropyridine receptor, which are called as the dihydropyridine receptors. Whereas on the T tubules, this is what is there in the T tubules. Whereas on the L tubules, I'm just drawing it below for seeing it properly, there is presence of a tubule that is not voltage gated. This on the L tubule, there is presence of the calcium channel which are not voltage gated. They are the ryanodine receptors of calcium. They are calcium ryanodine receptors which is not voltage gated. And why it is ryanodine? Because it opens by the plant alkaloid called ryanodine. Okay, this is the tubular system. Now, the triad, like if we have to draw the triad. So, here, let's draw the structure again. So, this is the, in a short form to understand, if this is a T tubule, then there are this dilation on the either side of this T tubule. This T tubule. Now, the dilation is called the terminal systole. Now, this whole unit, like the T tubules and the longitudinal tubules on either side, they are 3 in number because in between there is transverse tubule and other side on the either 2 side are the L tubule that is flanking them. The whole triad, this 3 structure together is called as triad. Now, this triad is most developed in the skeletal muscle, whereas in cardiac muscle, it is a triad. It is not a triad. Okay. Now, this triad, why it is important? Because what is the physiological importance of it? What is the physiological physiological significance of a triad. The physiological significance is that rather we should remember it for contraction of skeletal muscle, the calcium enters to the sarcoplasm from the triad, because without calcium there cannot be contraction of the skeletal muscle. So for the contraction of the skeletal muscle, calcium is a necessity, absolute necessity, and it enters from the triad. That is the physiological significance of the triad. The muscle fiber, it is made up of various muscle protein. So what are the muscle proteins? Now, muscle protein dependent on its function and location are divided into four types. Four types. The first one is anchoring protein, which is very important, because this is the one which is giving the mechanical stability to the muscle fiber when there is contraction and relaxation. Now, contraction and relaxation is a very strong process. Tension is generated, heat is generated, energy is required. So to maintain that, we have seen, right, there is like a pulling of the actin molecule towards the myosin during contraction. The sarcomere is like becoming smaller in length. So there is a mechanical process involved. So for the mechanical stability of this skeletal muscle fiber, this anchoring protein are very important, because all the muscles are elastic in nature. So it should not happen that the elasticity is lost or damaged, or the muscle tears off from the tendon. So for all this thing, the anchoring protein play a very important role. Okay. So anchoring proteins are Cap, Z, and actinin. Now, what is CAPC, or what are CAPC and actinin? Now, we know what are the lines. We know Z line, M line, H line, all the lines we know. So, these proteins anchor the M line that runs between the A band to the Z line of the sarcomere. That is a CAPC and actinin. That is their importance. Okay. Second is titin. The titin is also called the connectin. Now, this is a large protein that connects the Z line only to M line. So here we use a term only. In case of actinin, there is also an additional function, like it connects the M line to the Z line. Apart from that, also actin, that's why the name actinin, also actinin connects Z line to actin, that is the I band. But in case of titin or connectin, this is a large protein and it connects the Z line only to the M line, unlike the actinin and cap Z. Okay. Third is the this mean? Now, this is an intermediate filament. There are thick filament, thin filament, and intermediate filament in the cytoplasm. This jasmine is a intermediate filament, and we just mentioned a thin filament. What was that? The thin filament we mentioned is the actin. The thick filament that we mentioned is the myosin. So this means it is a intermediate filament. What it does? It stabilizes the scaffolds of the muscle proteins. Stabilizes the scaffolding of the muscle. We have myosin. It is present in the M line. Like whenever there is contraction, the M line bulges little bit. So the integrity should not get torn out. That is maintained by the myosin. Then next we have is the tropomoduli. This maintains the elasticity of actin in the center. And another important protein is the nebulin. Now, nebulin, what it does, it maintains the elasticity of actin throughout the length. Like from corner to corner, it maintains the elasticity. DGA, that is dystrophin glycogen complex. What does it do? These are the membrane stabilizing protein. Okay, these are the various proteins of the muscle which are anchoring in nature. The first one, the anchoring protein. Coming to the second protein, that is the second type of protein, which is the contractile proteins. What are the contractile proteins? The contractile proteins are the actin protein, which is thin filament. Now, actin protein, how they lie? They are first as monomer, then soon they form dimer. In monomer form, they are called as the G-actin. Then they form the dimer. From there, they form the F-actin. Now, it is the F-actin that polymerizes, that undergo polymerization. Polymerization F-actin. Now, this is this polymerized F-actin that is a mature actin filament. Okay, this is the actin. The second regulated, this contractile protein is the myosin. Now, myosin, it has three parts. It is also the thick filament. The myosin has three parts. What is its three parts? Head, and then there is a flexible neck, and there is a tail. So, what is the structure of a myosin? A typical structure of a myosin is like this. Typical structure of a myosin is something like this. It is a double helical structure. This helical structure, these are called as the heavy chains. These are called as the heavy chains. Now, from here extends the flexible neck, and this is the tail. The neck portion has the lobular head. It has this lobular head. Now, this globular head has two myosin light chain. They have myosin light chains, and the site they have, there is like one ATP binding site. One is the ATP binding site. Another one is the myosin binding site or regulatory site. So, ATP binding site is also called as the catalytic site because the ATP binding site has ATPase which can break down the ATP. So, one is the catalytic site on the ATP binding site, another one is a myosin binding site or the regulatory site. This is the structure of a myosin. Now, myosin is usually 1 to 6 microns in length, and myosin 2, there are like 6 subunits of myosin 1 to 6. Myosin 2 is present in the skeletal muscle. It is the myosin 2 that is present in the skeletal muscle. Okay. The third type of muscle protein, that is regulatory proteins. These are regulatory proteins, like which regulates the contraction and relaxation. What means it regulates the contraction and relaxation? It means it will control or decide the binding of actin to myosin. These are troponin. It has three subunits. It has three subunits like I, T, and C. So, I is the inhibitory site or unit. T, it is the tropomyosin binding site, and C, it is the calcium binding site. Okay, these are the three units. So, this troponin I subunit, it covers the binding site on the actin where the myosin should bind. T binds to the tropomyosin, and C subunit is the site where the calcium will bind. Next regulatory molecule is tropomyosin. Now, tropomyosin, what it does, it groups the actin molecule. So, if we are to draw that one, this is like tropomyosin, and on it we find the actin and troponin. Okay, this is how the tropomyosin groups the actin like, and here lies the troponins. So this is the troponin, this is the actin, and this is the troponin. So these are our regulatory protein which are present in the muscle. Now coming to the relaxation proteins of the muscle. So what are the relaxation proteins of the muscle? Like, it is true that calcium and the binding and unbinding of this actin and myosin carries out the contraction and relaxation. But additionally, there are the proteins which are pumped in nature who remove the excess calcium so that in presence of calcium, a premature contraction should not happen. I repeat, there are pumps which removes the calcium which is present in the cytoplasm, that is sarcoplasm, after the completion of contraction of a skeletal muscle. Because if excess calcium is present, then before time there are chances of triggering another contraction of muscle, which would lead to like spasm like condition, tetanus like condition. So the relaxation proteins are the sarcapum. It sequesters, means remove. It sequesters calcium from sarcoplasm after contraction. Okay, sarcapum. Another is calsequestrine. The calsequestrine, this pump buffers the calcium in sarcoplasm after contraction. Okay, these are the various type of the muscle protein. First was the anchoring proteins, contractile proteins, regulatory protein, and then the relaxation protein. These are our. Next is understanding the neuromuscular junction. Now, what is a neuromuscular junction? Now, neuromuscular junction, this is a special synapse. It is a special synapse in which, this is a special synapse in which the alpha motor neuron innervates a skeletal muscle fiber. Here, one alpha motor neuron innervates a skeletal muscle fiber because muscle to contract, there has to be one innervation, and that innervation comes from the motor neuron which is originating from the spinal cord. So, alpha type of spinal motor neuron is the one that goes and supplies the skeletal muscle fiber. Now, if we are to draw this one, it would be like if this is the axon, terminal part of the axon, the synaptic knob. It is going and innervating the muscle fiber. So, the first structure that will come in contact, it is the sarcolemma. It is the sarcolemma. Okay, in a simplified manner, we are drawing the diagram. So, this is the axon terminal and this is the sarcolemma and this is the synapse. So, in a neuromuscular junction, there are three parts. What are the three parts in a neuromuscular junction? In a neuromuscular junction, this is the synapse, like the contact point between the muscle fiber and the alpha motor neuron is called as the synapse. Okay. Now, if this is the synapse, so the area before the synapse is the presynaptic region. And the area after the synapse, it is the postsynaptic region. Now, in the presynaptic region, what is happening? What is the event that is happening? We know the nerve, it has the capacity to generate impulse, right? It generates impulse and it flows from the axon here out. So, when the wave of depolarization, this is wave of depolarization. So, when a wave of depolarization reaches the axon terminal, now in this axon terminal, as soon as it reaches, what it does immediately, it will cause influx of calcium. It will cause influx of calcium via the calcium voltage gated channel. Now, once this calcium comes into the sarcoplasm, what it will immediately do? It will form acetyl-CoA. It will form acetyl-CoA via the acetyl-CoA transferase. Acetyl-CoA transferase. It will form the acetyl-CoA. Now, this acetyl-CoA within the synaptic knob, it will be lysed into the synapse, which is also called as the synaptic cleft of the synapse. Now, in the synaptic cleft, that gap between the alpha motor neuron and the muscle fiber membrane, that is the sarcolemma. What is there in the postsynaptic region? In the postsynaptic region, there are this receptor for this acetyl-CoA. So, this acetyl-CoA it will bind into its NM type receptor. Once this acetyl-CoA is bound to the NM type of receptor, what it will do? It will cause sodium influx. It will cause sodium influx. Now, we know there are this ryanodine receptor as well as dihydropyridine receptor. Now, this sodium influx is what? Sodium influx is generation of the wave of depolarization. So, when from the neuron, the wave of depolarization via acetyl-CoA, because acetyl-CoA is an excitatory neurotransmitter. So, acetyl-CoA, after it binds onto the receptor in the postsynaptic region, it will again induce depolarization wave in the membrane of the muscle. Now, this will open the dihydropyridine receptor through which calcium will enter. Now, this voltage gated calcium channel, it will induce configurational change in the ryanodine receptor. So, as a result of which calcium will be pulled out of the sarcoplasmic reticulum into the sarcoplasm, and then the contraction of muscle will happen. So, very briefly I repeat, then we will move on to the other parts of it, like the wave of depolarization from the neuron across or along the exoplasm, it comes to the axon terminal. When it comes to the axon terminal, it opens the voltage gated calcium channel. When the voltage gated calcium channel is opened, immediately acetyl-CoA is formed from by acetyl-CoA transferase. Now, this formed acetyl-CoA through synaptic vesicle is released into the synaptic cleft by the process of endocytosis. Exocytosis. So, by the process of exocytosis, this calcium-induced acetylcholine is released into the synaptic cleft. So, as soon as it is released into the synaptic cleft, it binds onto the NM receptor of the acetylcholine. It is NM receptor for the acetylcholine receptor. Now, when it binds, it is a excitatory neurotransmitter. It will induce wave of depolarization. First, calcium will enter through the dihydropyridine because it's a voltage gated channel, which again will induce changes in the ryanodine receptor, and more calcium will enter into the sarcoplasmic reticulum. Thus, there will be wave of depolarization. Now, coming on to the applied part. Here, after this acetylcholine does its function, still there are certain molecules of acetylcholine left, which is digested by the acetylcholine esterase that is present in the synaptic cleft. Now, this acetylcholine receptor, it is supported by a a cluster of protein which are like 12 in number, like rhabsine. Then you have the agrine. Then there is this docking protein which is called the DOC7, and then there is additional low protein lipoprotein receptor ILP4 ILPR4, and another important stabilizing protein that is also present to support this acetylcholine structurally is the mask protein. Okay, now the whole process, writing it in flow chart so that we can connect to the applied part and clinical part. So, it is depolarization of axon opens voltage gated calcium channels in the axon terminal. Okay, acetyl CoA is formed via the synaptic vesicle exocytosis of calcium occur in the synaptic cleft. Now here one thing you have to remember, after this acetylcholine is released in the synaptic cleft, there is wave of depolarization. Now as the this is called as the end late potential. Even during resting stage, when there is no wave of depolarization, still there is little bit of acetylcholine that causes small waves of contraction of the skeletal muscle. That's why we have the tightness of the skeletal muscle. So after this end plate potential is generated, acetyl CoA binds on the presynaptic membrane acetylcholine receptor, which is the NM type. Okay. Now, this acetylcholine that binds on the acetylcholine receptor, acetylcholine is a excitatory neurotransmitter. That means it will propagate, it will pass the wave of depolarization further into the muscle. So, muscle membrane undergoes depolarization. Sodium influx in the sarcolemma. What does it cause? Opening causes opening of dihydropyridine receptor, which is voltage gated calcium channel. It induces opening of the ryanodine receptor and movement, or rather influx of calcium from the terminal cisternae, which is a part of the triad. Terminal cisternae, like as we have mentioned, see here this dihydropyridine and ryanodine receptor, they are present in the T tubule and the L tubule respectively, right? So the role of the triad is here. This is the role of the triad. A movement or influx of the calcium from the terminal cisternae into the sarcoplasm happens, resulting in contraction of, okay, this is what is the neuromuscular junction, and it's the events that is happening in the neuromuscular junction. Now, when this all are over, then the acetylcholine esterase, okay, removes the excess acetylcholine from the synaptic cleft. Okay? This is the function of the acetylcholinesterase. Now, clinically, what is this neuromuscular junction used or implied? What is its clinical implication? Okay. Now, neuromuscular junction is the site for drugs to act. Okay, like to carry out certain surgery very quickly, sometimes we induced prolonged depolarization or we induce like relaxation. So there are certain drugs that target the neuromuscular junction and cause like sustained depolarization or they stop the depolarization. Now this is of two types. Neuromuscular junction can be inhibited in two ways. Means the passage of the impulse will be inhibited in two ways. Okay, to the muscle. Like first is the non-depolarization or competitive inhibition. Non-depolarization or competitive inhibition. Now here drugs like tubocurarine, drugs like tubocurarine binds on the acetylcholine receptor and blocks the binding of acetylcholine on acetylcholine receptor. Thus, if those drugs bind on the receptor of the acetylcholine, so there is no place for the acetylcholine to bind on its receptor because it is already bound. It has competitively inhibited the acetylcholine from binding onto its receptor. Thus, no depolarization occurs. This is one. Second type is that the drugs that kind of mimics the function of acetylcholine, like depolarizing or non sorry, which is like depolarizing, non-depolarizing. The non-depolarizing was a competitive inhibition. Now, non-depolarizing will not happen here. Rather, depolarizing will be induced. It will mimic the function of the acetylcholine. So, depolarizing and non-competitive inhibition means here this drug it will not compete to bind onto the acetylcholine receptor. Rather, it will exaggerate, it will mimic the action of the acetylcholine. So, example succinylcholine. Now, succinylcholine is very much same like that of the acetylcholine. So, what it will do? It will induce prolonged depolarization on the postsynaptic membrane. Now, the advantage that this has is that here the advantage is there is no esterase to digest succinylcholine. Its effect just goes away, wears off after 5 to 10 minutes. Okay, this is the two type by which the neuromuscular junction is targeted. One is by non-depolarizing method, another is a depolarizing method. Some drugs like neostigmine, they target the acetylcholine esterase. Now, maybe due to some congenital condition, there are diseases of the neuromuscular junction like myasthenia gravis, Lambert-Eaton syndrome, all this thing in which there is not enough acetylcholine available for the depolarization. So, in that condition, the drugs like neostigmine is used to block the activity of the acetylcholine esterase. So if acetylcholine esterase is not there, then whatever acetylcholine is available or released in the synaptic cleft, it will get enough time to pass on the wave of depolarization onto the sarcolemma. So that is the reason why to treat condition where there is not enough acetylcholine, drug like neostigmine is administered, which will block the activity of the acetylcholine esterase. Okay, now there are certain toxic substances also that are used to target the neuromuscular junction, but their site of action is different. Like drugs used to block various channels in the neuromuscular junction. Example, tetrodotoxin. What does tetrodotoxin does? It blocks the sodium channel in the pre in the presynaptic membrane. Now, this tetrodotoxin, it is a toxin from a puffer fish. It will block the voltage-gated sodium channel. Second, we have the dendrotoxin. This is a toxic substance from the mamba snake. So, what it does, it blocks the potassium channel on the presynaptic region, that is on the axon terminal. Next we have is the conotoxin. It is a toxic substance that is derived from the snail. So this conotoxin, it locks the calcium channel on the presynaptic region. Okay, so we have understood the neuromuscular junction and we have also understood its clinical application. So from this video, what did we learn? We learned about the salient structure of the muscle fiber. We had a conceptual understanding of the sarcomere and how is it applied. We understood the concept of sarcotubular reticulum system as well as the triad. Then we gained knowledge about the various muscle proteins in context to their physiological function. At last, we had a vivid idea about the neuromuscular junction and the clinical aspect related to neuromuscular junction. Okay, so I will again see you tomorrow. Till then, thank you.